Hi, everyone. I'm Krista Power from the University of Ottawa, and I'm here to give you a short presentation on the interplay between diet and the microbiota and colon cancer. So I'm just going to share my screen and switch over so you can see my presentation. Okay. So today's lecture, we're going to cover just a little brief introduction of the microbiota and its links in colon cancer, and then look at the interplay between the diet and the microbiota in colon cancer prevention. Looking at components of the diet that have preventative or therapeutic effects, such as the non-digestible carbohydrates, as well as a brief intro into certain phytochemicals and their role in preventing colon cancer. And then we'll end today's session with some examples of components of the diet that can actually be cancer promoting, so certain dietary proteins as well as dietary fat. So we know that colon cancer is the third most common cancer type in Canada and is one of the leading global causes of cancer-related deaths. So shown here in this table are the new cases in Canada of all cancer types in males and females. And as you can see, colorectal cancer represents 13% of all new cases in 2019 in males and 11% in females, which is below prostate and lung or breast and lung in women. So there are some cases of colorectal cancer that have been linked to inherited germline mutations. So that makes up about 25% of the colon cancer cases, but the vast majority are sporadic carcinomas, which are devoid of any familial or inherited predispositions. So that suggests that there's other causes outside of genetics that can drive colon cancer development. And that includes a number of environmental factors such as diet, sedentary lifestyle, as well as obesity and aging. And also intestinal inflammation has been linked to increases in colon cancer development. As it's known, individuals who have inflammatory bowel disease, for example, have increased risk of developing colon cancer. But what we're going to talk about today is the role of microbial dysbiosis. So that's a change in the composition of the microbial community and the function of that community that's different from healthy individuals, which is microbial dysbiosis is known to be high in subjects with colon cancer and those that are at risk of developing colon cancer. And we're really going to look at the relationship between diet and the microbiota as it relates to colon cancer. So the human microbiota consists of 10 to 100 trillion microbial cells, which includes bacteria, fungi, and viruses. And as we're moving through discussions with colon cancer, we're really going to be focusing on bacteria. But the microbiota has been associated with approximately 20% of the global cancer burden, which has been associated with specific microbiota. So you can see that with cervical cancer and certain viruses, as well as gastric cancers. And Helicobacter pylori, for example, is known to be associated with gastric cancers. But when it comes to the colon and colorectal cancer, it's not really just one microbiota that's associated, but really a change in the composition and the function of the microbiota. And the interest in really investigating which parts of the microbial community are associated with colon cancer has been driven because the colon houses the greatest proportion of the bacterial community in the human body. So comparing to different sites, and as well, there's a 12-fold increase in cancer risk as you move further down the intestinal tract. So in the large intestine compared to the small intestine, there's an increase risk of developing cancer, which is suggestive that the microbiota may be playing a role in driving colon cancer development. Also, there's evidence in animal models that are suggestive of a link between the microbiota and colon cancer. So in mice that have been germ-free, so they technically do not have a microbiota, they have less carcinogen-induced tumors compared to mice that have a normal microbiota, suggesting that not having a microbiota is actually protective, and vice versa. There's things in the microbiota that may be driving cancer development. Similarly, in mice that are genetically susceptible to colon cancer development, when they're germ-free, they develop significantly fewer tumors compared to their counterparts that have a microbiota. So collectively, it's really pointing to a role of the microbiota in driving colon cancer development. So we know that the microbiota is composed of different phyla. So the main phyla in the colon and small intestines would be the firmicutes and Bacteroidetes, as well as smaller proportions coming from proteobacteria, fusobacteria, and actinobacteria. So between the colon and the small intestine, the proportions of these are basically stable. However, the individual members that are residing in the different areas of the intestine will change depending on the location. And we know that in healthy subjects, so a study was conducted where they looked at the microbial composition between colorectal cancer patients, patients that have high susceptibility of developing colon cancer, as well as healthy subjects. And they noticed that the composition or the community structure in the subjects that were healthy were different from those that were either highly susceptible to developing colon cancer or those with colon cancer, which is suggestive that the community has changed during the development of colon cancer, which is reflective here. And looking further to see what those changes are, studies have looked at the microbiota in more detail, and they've been able to show that compared to cancer-free subjects, those with colorectal cancer have higher species richness, for example. They have lower abundance of potentially protective taxa, such as Roseburia, and they have increased abundance of pro-carcinogenic taxa, such as Bacteroides, Escherichia coli, for example, and the Fusobacterium. So Fusobacterium would be within this phylum here, and it's been shown in a number of colon cancer in studies that have been profiling colon cancer that this phylum is actually increased in subjects with colon cancer compared to healthy controls. So showing that the composition of the microbiota itself is altered or dysbiotic in colon cancer patients. So there's a number of mechanisms that can be associated with the microbiota and colon cancer, and we're not going to cover all of them today. I'm just going to give you a brief summary. So the microbiota itself, when it's dysbiotic, they can trigger a number of inflammatory pathways that can increase inflammation in the epithelium of the colon, which can drive colon cancer development. Bacteria also have a number of enzymes that have been shown to activate different procarcinogens that can induce some oxidative damage, oxidative stress, which can lead to DNA damage, altering, ultimately increasing colon cancer development. All of these changes as well are known to actually increase inflammation in the intestinal lining, for example, which can cause damage or reduce the integrity of that barrier, which can further perpetuate this inflammatory cascade. So we're going to look at how diet can actually play a role in either reducing some of these pathways known to, that the microbiota can use to drive colon cancer development, or which can aid in or cause an increase in the microbiota driving colon cancer development. So we look at the interaction between the diet, microbiota, and colon cancer. So we know that different food components can have specific effects on the microbiota, so they can change the composition, which members are there, how many of them are there. The diet components can also have effects directly on the intestinal mucosa, either promoting barrier integrity or having some adverse effects on the integrity of that barrier. And so the communication between all of these components can either increase the risk of colon cancer development or alter the behavior of tumors, or in the case where it's protective, then it can reduce colon cancer risk. So we're just going to go through a few examples here of different food components that can really have a role in modulating this interaction between the microbiota and colon cancer. The first is dietary fiber. So dietary fiber, which are ultimately the components of our diets that are carbohydrate in origin, but they're non-digestible, and they're known to play an important role in colon health in general, and also in colon cancer prevention. So when we look at carbohydrates coming from our diet, there's really two main types. You have your simple sugars and starch, which are digested and absorbed in the upper gut or the small intestine. And then you have your dietary fibers, which are non-starch polysaccharides, oligosaccharides, and resistant starch. So these are complex carbohydrates that escape or are resistant to the digestive enzymes of the small intestine, resulting in their entry into the colon, and therefore their availability to the microbiota. And these dietary fibers, it's a huge class of compounds, so it's not just one thing, but they're high in plant-based foods. So the cereals and grains, as well as the legumes, such as dietary pulses and different seeds and vegetables, have a rich array of different dietary fibers. And overall, it's known that dietary fiber can increase viscosity and fecal bulking in the colon. So this ultimately has a diluting potential on carcinogens, so restricting the accessibility of carcinogens to the colon. They're also known to bind potential carcinogens, such as secondary bile acids, which we'll talk about towards the end of the lecture, so therefore making them unavailable to act negatively on the colon barrier. Dietary fiber is also known to lower the luminal pH, and this is really related to its ability to act as substrates for microbial fermentation. So dietary fiber can reduce luminal pH, making it a more acidic environment, which will alter which microbiota are able to grow in that environment. So we'll look more closely at the role of dietary fiber as substrates for microbial fermentation, because it plays one of the most important roles in dietary fiber's preventative effects on colon cancer. So certain classes of dietary fibers are fermentable, and so they're fermented by the microbiota to produce short-chain fatty acids. That includes acetate, propionate, and butyrate. Usually, in most situations, acetate is the most abundant short-chain fatty acid, followed by butyrate and propionate, which are usually present in equal amounts. However, that really depends on the type of fiber that's being consumed, as well as which microbiota are actually there, because it's the microbiota that possess the enzymes that are capable of converting the dietary fibers into the short-chain fatty acids. So it's really dependent on fiber type and the microbial composition. And the degree to which the dietary fiber is fermented is dependent on the physiochemical properties of fiber itself. So what the structure of the fiber is, whether it's soluble. So typically we think of soluble dietary fibers as being fermentable, whereas insoluble dietary fibers are not fermentable, so they wouldn't be able to produce these short-chain fatty acids. So fiber sources from oats, dietary pulses, and flaxseed are highly fermentable. So they're rich in dietary fibers, soluble dietary fibers such as pectin, arabinoxylands, as well as mucilage. Whereas cellulose, which is an insoluble dietary fiber from wheat bran, are poorly fermentable. So you wouldn't have a lot of short chain fatty acids being produced upon consumption of cellulose. So the link between short chain fatty acids and colon cancer really come from butyrate. So this is the short chain fatty acid that has direct effects in the colon itself, whereas acetate and propionate would be more absorbed and used systemically. So, sorry, my dog's about to bark at the cat, so I'll try to calm him. So when we look at short chain fatty acids, it's known that they can decrease colonic pH, thereby decreasing the growth of pathogenic bacteria, which do not like an acidic environment. Therefore, it supports the growth of healthy commensals. Butyrate is also an important energy source for colonocytes, so it induces a tropic effect. So that's just another word for increases the growth or the health of the colon epithelium. Short chain fatty acids, in particular butyrate, are also known to enhance intestinal barrier function, and this is through its ability to increase the expression of tight junction proteins, which are really the proteins that keep the epithelial cells together. So it plays a role in promoting barrier integrity or reducing barrier permeability. They're also known to enhance colonic microbial defense responses, so stimulating mucus secretion, for example, thereby protecting the colon epithelium from being in direct contact with the microbiota. And also, because of its effect on enhancing barrier integrity, it can also reduce bacterial translocation into the colon barrier, which would stimulate inflammation and drive colon cancer development. Short chain fatty acids are also known to have anti-inflammatory effects, so they can inhibit NF-kappa B signaling. They act through their respective short-chain fatty acid receptors to promote regulatory T cell differentiation, resulting in an increase in anti-inflammatory cytokine production. And we know that in subjects with inflammatory bowel disease, they actually have a defective butyrate metabolism. So this is thought to be one of the reasons why why IBD patients have so much inflammation. It's due to the lack of butyrate utilization. We also know that butyrate treatment can reduce inflammation in IBD patients, so suggesting a role of increasing fiber consumption in IBD patients as a treatment. Short chain fatty acids have also been shown to induce apoptosis and cell cycle arrest in colon cancer cells. So these other features were really on the preventative side, but on a intervention side, short chain fatty acids can reduce colon cancer proliferation. They're also known to reduce oxidative stress and DNA damage and enhance the endogenous antioxidant defense system. So overall, fiber, through its production of short chain fatty acids, in particular butyrate, can have a preventative and a therapeutic role in colon cancer. In addition to increasing the activity or increasing the production of metabolites by the microbiota that can have beneficial effects on different stages of colon cancer development, fiber is also known to change the composition of the microbiota, so which members are present in that community. So this has been done in population studies, so observational studies where they look at the microbiota of populations that have high fiber diets, and they know that they have low levels of Firmicutes and increased Bacteroidetes, in particular microbiota that are known to be saccharolytic. So they're responsible for metabolizing fiber to produce short chain fatty acids. And in more controlled clinical trials, evidence suggests that feeding resistant starch, for example, the class of dietary fiber, can increase short-chain fatty acid production and bacteria that can degrade dietary fiber. And also treatment with different prebiotics. Inulin, for example, which is present in onions, garlic, and asparagus, has been shown to stimulate the growth, again, of different microbial species that are known to produce short-chain fatty acids. So not only the end result of microbial fermentation of fiber resulting in short-chain fatty acids can have beneficial effects, but fiber itself can change the composition of the microbiota into a more beneficial community structure. So in addition to dietary fiber, there's also some dietary phytochemicals that are known to be protective against colon cancer, for example. So I brought up these phytochemicals as an example because they have a specific relationship with the microbiota that increases their activity. So the phytochemicals, there's thousands of them. They're present in small abundance, but really diverse in a number of plant foods. Soy, asparagus, berries, flaxseed contain these phytochemicals. And they're divided into different classes. So there's flavonoids as well as the lignans. So what makes them very unique is that they are degraded or metabolized in the intestinal tract, but they're further metabolized by the gut microbiota, and the microbial metabolites of these phytochemicals are actually more bioactive. So it's beneficial when the microbiota can convert these phytochemicals into their more bioactive metabolites. So, for example, with genistein, which is present in soy, the microbiota can convert it to Equal, which is very potent anti-inflammatory component. Similarly, quercetin is a microbial metabolite of rutin, which is rich in asparagus. So rutin itself doesn't have very much biological activity, but when it's converted by the microbiota into quercetin, you have a lot of anti-inflammatory effects. Similarly, with flaxseed, it contains the richest source of plant lignans, which again have minimal biological activity, but when they're converted into the mammalian lignans by the microbiota, you can see the activity of these components increase. So when we look at the relationship between these microbial metabolites of the phytochemicals, we can see that they have effects on modulating different enzymes. They have anti-inflammatory effects, and they've also been shown to reduce cell proliferation and increase apoptosis, all of which have been linked to a reduction in colon cancer development. So we'll move on now into looking at some of the components of our diet that can have adverse effects on this microbiota colon cancer relationship. So we'll look at components that are present in meat, for example. So we know that amongst all cancers, colorectal cancer is most strongly associated with meat consumption. Meat consumption itself, if it's consumed in moderation, does have beneficial components. So it's an important source of protein and different nutrients in our diet, including iron, zinc, and vitamin B12. But we know that increased consumption of meat, specifically red meat and processed meat, can increase colon cancer risk in a dose-dependent manner. You can see here in this forest plot, they're comparing colon cancer risk in subjects who have high meat consumption compared to those who have low meat consumption. And you can see the risk of colon cancer development increases in subjects who are consuming high levels of red meat as well as high levels of processed meat. And it's known that the risk of colorectal cancer increases by 30% with every 100-gram increase per day of red meat consumption, and 21% for every 50-gram per day increase in processed meat consumption. So just showing you here what 100 grams of red meat would look like. So just it fits in your palm of your hand. So for every increase of 100 grams per day increases your risk of developing colorectal cancer by 30%. So there's a number of components that are known to be linked to colon cancer development with regards to meat consumption, but we're just going to focus today on the ones that may be associated with the microbiota. And the first one would be protein. So really, when dietary proteins are ingested, they are typically metabolized, digested and absorbed via enzymes within the small intestine. So these would hydrolyze your proteins into small peptides and into amino acids, which would then be absorbed and utilized by the body. But when you have decreases in enzyme function, for example, with aging or increased load of protein going into the body, you can have some dietary protein that escapes digestion and absorption, and therefore can be enter into the colon, where it can be acted on by the microbiota. So the microbiota also possesses enzymes that can hydrolyze and metabolize dietary protein that enters into the colon. And specific bacterial members have been shown to have high proteolytic activity. And importantly, these enzymes, these proteases, are more active at neutral to slightly alkaline pH, which is in the distal colon. And this is important because most of the colon cancer cases are in the distal region with that high pH compared to the proximal colon, which has more acidic pH. So what happens when the microbiota are exposed to proteins in the colon? Well, we know after proteins are hydrolyzed, they turn into amino acids, and amino acids then can be acted on by the microbiota in order to produce a number of metabolites. And this really depends on the type of amino acids that are present. So you can see here, we can have sulfur amino acids, which are converted into hydrogen sulfide by the microbiota, which is a cytotoxic substance, which I'll discuss in a couple of more slides about how hydrogen sulfide is cytotoxic. You can also have deamination and fermentation, which produces, again, short-chain fatty acids and branched-chain fatty acids. And these branched-chain fatty acids are coming from the branched-chain amino acids, so leucine, isoleucine, and valine. And there's really no known adverse effects of this pathway with regards to microbial metabolism of the amino acids. But these other ones do have known adverse effects in the colon. So ammonia, for example, is known to inhibit short-chain fatty acid oxidation, can alter cell proliferation, have deleterious effects on the intestinal barrier, thereby reducing the integrity, and is also known to increase tumor incidence in different animal models. So the more protein that comes into amino acids can result in an increase in ammonia, which is deleterious. You can also have microbial activity on the aromatic amino acids to induce phenols and indols. So this would result in phenylalanine going to a phenylacetic acid, tyrosine producing P-cresol, and tryptophan being converted into indol. And we know that phenol is a co-carcinogen by enhancing nitrosation of secondary amines, and it also enhances epithelial barrier permeability, so it has deleterious effects on the integrity of that intestinal barrier, which can lead to an increase in inflammation and enhancing the carcinogenic cascade, for example. Indole also has been shown to reduce gut barrier integrity and also has inhibitory effects on the survival of different probiotic species. So microbial species that are known, for example, to produce short chain fatty acids. Indole is known to reduce their survival. So overall, increasing the amount of protein and specific proteins, depending on the amino acid structure, can result in a number of deleterious microbial-derived metabolites that can have adverse effects in the colon, leading to an increase in colon cancer development. So there has been some work in the past looking at different types of protein and their effects on the development of colon cancer or events that can lead to an increase in colon cancer risk. So they look at the colon barrier integrity, for example, where they studied the mucus layer thickness. So it's just a measure of how healthy that epithelial barrier is that separates the microbiota from the colon, as well as DNA damage, which is one of the driving forces for colon cancer development. And so what they did here, this is a rat study. So they fed rats different diets containing either 15 or 25% of protein coming from different sources. So casein and whey are two milk proteins, and they also looked at soy protein. And here you can see the 15% and then the 25% and similar for the order in this figure. So what you can see here is that when you increase the amount of protein, you increase the amount of DNA damage in the colon of these animals. And this was true for both casein and for soy protein, whereas whey, which is the protein source from milk, that's more digestible, so you would have less coming into the colon. There was very little effect. And also, when they looked at the mucus layer thickness, they could see with casein, not so much with the other protein types, but it had a significant effect on reducing the thickness of that mucosal barrier. So really would enhance the accessibility of the microbiota to the colon barrier that can induce a lot of inflammation. So really showing that the effect of the microbiota, the effect of the protein on the colon is specific to the protein type as well as concentration. What was interesting in this study too is that when they combined these diets with a source of resistant starch, they were able to attenuate the damage that could be done by increasing the protein content. So you can see here when resistant starch was added to the diet, so that's a type of dietary fiber, you could attenuate the DNA damage that was done by increasing casein as well as soy in the diet. So this effect was attenuated. Similarly, with the mucosal layer thickness, adding resistant starch to the diet prevented this deleterious effect caused by increasing casein concentration in the diet. So ultimately, the quantity and type of protein influence the risk of colorectal cancer development, which can be modified by fermentable carbohydrates. So really having a balanced diet of moderate amount of protein with more fermentable carbohydrates could help attenuate any of the damage that could be caused by increased protein load to the colon and interaction with the microbiota. So the last dietary component that we'll talk about with regards to its link to enhancing colon cancer risk and development is dietary fat. So a number of studies have shown that total dietary fat, or in particular saturated dietary fat, has a positive association with colon cancer incidence. And this is demonstrated in a number of studies, but is not consistent throughout. We also know that the type of fat is important because these associations are not linked to polyunsaturated fat intake, especially those of the N3, which are rich in fish, for example. So it's also been demonstrated in a number of animal models. So this mouse model is highly susceptible to developing colon cancer, for example. And when these diets are fed a high-fat diet, it can increase the risk of tumor development. or not the risk, it's significantly increased tumor development. So just showing here in this study, so this is the diet composition of their normal diet, where they had 15% fat making up that diet, and when they gave the high fat diet, they increased the fat level to 45%, and this was in the form of saturated fats. And you can see here with the total tumor number, it basically doubled in the animals that were fed a high fat diet, showing a relationship between the fat content and type on tumor development. This is not only shown in animal models that are susceptible to colon cancer, but also in normal mice, giving them a Western style diet, excuse me, has been shown, like, that have elevated fat and decreased calcium and vitamin D, have also been shown to have increased tumor numbers after consuming these diets for a prolonged period. And there's a number of mechanisms through which dietary fat can influence colon cancer development, but I'll only talk about now the ones that are linked to the microbiota, and this is really related to the impact of microbial-derived secondary bile acids. So we know that bile acids are necessary for dietary fat absorption. So this starts with the primary bile acids, so cholic acid and chenodeoxycholic acid, which are synthesized in the liver from cholesterol. They're conjugated to amino acids, taurine or glycine, and then they're carried into with the bile into the small intestine, where they aid in the digestion and solubilizing and digesting and absorbing dietary fat. So really, when you're consuming a lot of fat, these bile acids are delivered to the small intestine, where they aid in absorption, and then they're basically reshuttled back from the small intestine into the liver. So they're important for ensuring that cholesterol, dietary fats, fat-soluble vitamins from the small intestine are soluble and absorbed. And there's high concentrations of bile salts maintained in the different portions of the small intestine where fat digestion absorption takes place. And then bile salts are reabsorbed through passive diffusion and through high affinity active transport from the distal ileum. But you can see here that not all of the bile salts are taken away from the small intestine. So there's a small portion, depending on your diet composition, that can enter into the colon. And that's really where we're getting our link between dietary fat and the microbiota and colon cancer. So primary bile acids that reach the colon is only about 5%, but that could lead to a high amount depending on how much fat you're consuming. These are deconjugated and undergo enzymatic reactions to form secondary bile acids, which are produced solely by the actions of specific microbial enzymes. And that includes bile salt hydrolases, hydroxysteroid dehydrogenase, as well as 7-alpha dehydroxylase. And we know that this results in the conversion of cholic acid into secondary bile acid, deoxycholic acid. And this one has the most important links between dietary fat and colon cancer. You can also have secondary metabolites coming from chenodeoxycholic acid into lithocholic acid, which there's less evidence to suggest that this secondary bile acid has any links to colon cancer. And we know that in subjects with colon cancer or at high risk of developing colon cancer, there's an increased amount of these secondary bile acids present in the feces, which is really pointing towards a causal association between these secondary bile acids and colon cancer development. And we look at the mechanisms of how these secondary bile acids can impact colon cancer development. They're linked to the microbiota. Well, we know when they're consumed, we need the microbiota when they're delivered to the colon in order to deconjugate them. So specific enzymes, as I was mentioning, can deconjugate the primary bile acids, thereby removing its glycine and taurine. And these unconjugated bile acids then are acted on by other microbial enzymes that produce these secondary bile acids. And the secondary bile acids are known to alter the gut microbiota. So the composition resulting in dysbiotic microbiota, as well as promoting the increasing the enzyme activation of the enzyme responsible for converting the primary bile acids into the secondary bile acids. So really, it would perpetuate this cycle of increasing the production of secondary bile acids. These secondary bile acids are also known to have genotoxic metabolites produced, so it increases oxidative stress and DNA damage, thereby increasing the tumor promoting activity in the colon. So this diagram is just highlighting here that the secondary bile acids are really connected to the microbiota, which is driving their tumor promoting activity. It's also been demonstrated in different mice models. So in a mouse model that has a defect in the bile acid transporter. So this is the transporter that would have delivered or taken the bile acids out of the small intestine, and therefore decreasing how much would go into the colon. And these animals have increased carcinogen and inflammation-induced colon tumor development, really showing that increasing the load of the secondary bile acids in the colon is responsible for increasing the carcinogenicity of these secondary bile acids. And the last thing I want to talk about is this, the impact of this increase in taurine on this whole cycle. So it's not just the secondary bile acids that can have a negative effect, but also increase in taurine. So glycine and taurine serve as substrates in microbial metabolism. So there's certain bile-loving bacteria that can metabolize these amino acids. So taurine in particular is important because it has a sulfur-containing amino acid, and it's converted into hydrogen sulfide after deconjugation by the microbiota. And we know that hydrogen sulfide is highly toxic and linked to colon cancer because it's been shown to increase proliferation of colon cancer cells. It inhibits butyrate metabolism, inhibits apoptosis, so enhances the growth of tumors, and it also increases secondary bile acid conversion. So again, increasing that whole cycle. And we know in ulcerative colitis patients, for example, and it's a form of inflammatory bowel disease, they have defects in the ability of the colon to detoxify hydrogen sulfide. So it's really one of the links between why patients with IBD have increased risk of developing colon cancers, because they have high levels of hydrogen sulfide in the colon. So we know that looking at this in total, that meat-rich diets significantly increase both the level of taurine conjugated to bile acids and the production of hydrogen sulfide in the colon, thereby linking it to colon cancer development. So overall, looking at this pathway, it's not just the secondary bile acids that can have negative impacts on the microbiota, linking it to colon cancer development, but also the formation or freeing of this taurine that can increase hydrogen sulfide, further damaging, creating an unhealthy environment with the microbiota. Fortunately, again, as we were seeing with the negative impacts of increased dietary protein, dietary fiber as well as calcium have been shown to bind to secondary bile acids, increasing their excretion into the feces, and thereby can attenuate some of the adverse effects of these secondary bile acids in cancer development. So again, promoting a more diverse, balanced diet rich in dietary fiber can counteract some of the adverse effects of increased protein and fat in the colon. So just to summarize, we went through some of the effects that the microbiota have in the colon environment, increasing inflammation, increasing the expression of enzymes that can lead to pro-carcinogen activation and increasing colon cancer risk. But we also went through how the diet can interact with this whole cycle in order to either increase or decrease colon cancer risk. And more specifically, we looked at how the diet can alter the microbiota community structure and activity, how it can alter the barrier integrity and enhance in the with regards to the phytochemicals and fiber, but reduce in the situation where you have a lot of protein and fat in the diet. Can also have beneficial effects on modulating the immune and inflammatory response when it comes to dietary fiber and the phytochemicals, as well as reducing oxidative stress and DNA damage with fiber and the phytochemicals, and the opposite when it comes to fat and protein. And also reducing microbial exposure to carcinogenic compounds. So this is in relation to dietary fiber, for example, binding secondary bile acids that can have carcinogenic effects. So thanks for listening. I hope you're able to get a sense of how the diet and the microbiota really interact in either promoting or preventing or attenuating colon cancer risk. If you have any questions, my email is here. It's just krista.power@uottawa.ca. Be happy to chat about anything in this presentation. So thanks. See ya.